Hello, everyone. I'm Jeff Cummings, Professor of Brain Science and Director of the Center for Transformative Neuroscience at the University of Nevada, Las Vegas. It's my pleasure to welcome you on behalf of the Global CEO Initiative, the World Economic Forum, and the Davos Alzheimer's Collaborative to this Lausanne Dialogue on preparing the market for diagnostic innovations in Alzheimer's disease. Earlier this month, we reached a critical milestone in Alzheimer's disease, the approval of aducanumab. Regardless of how you feel about this approval, there is no controversy about the importance of amyloid PET in bringing us to this point in the dialogue regarding new therapeutics and new diagnostics. We could not want a more emphatic declaration of the importance of the topic we are considering today, biomarkers. Since the approval of amyloid PET approximately a decade ago by the FDA, we've seen continued innovation in diagnostic tools and diagnostic technologies across many modalities: blood, CSF, imaging, digital biomarkers, even breath. The innovation that we're seeing will help clinical trials and will help clinical diagnosis to eventually control Alzheimer's disease. Our first panel today, AD Diagnostic Innovation Pipeline, includes the experts and innovators behind these diagnostic tools and technologies. They'll share their insights on these innovations, tell us where they are in their innovation journey, and discuss the potential impact of their innovations on Alzheimer's disease. Our second panel, Opportunities and Barriers to AD Diagnostic Adoption and Scale, will address the opportunities and challenges of biomarker implementation, scale up, and reimbursement. The panel will explore how to improve the access to diagnosis and enable a timely, accurate, and compassionate diagnosis. So let's get started. For the first panel, I'd like to ask the panelists to turn on your screens. I think you all do, and offer a 2 to 3 minute introduction of yourself and of the innovation that you represent. Mike Weiner, let's start with you. Is Mike on with us? If not, Steve, could I ask you to start?
I'd be happy to, Jeff, and thank you and the organizers for inviting me here today. So I'm Steve Verduner. I'm CEO and co-founder of Neurovision Imaging. We're a company that's developing diagnostic solutions in the neurodegenerative space. We work in concert with pharmaceutical companies for about 10 years now. Certainly a challenging space, and yes, it's good to see an approval regardless of your position on it in driving the field forward. So in the couple minutes that I have, I'd like to tell you a little bit about our company and the two major initiatives that we have. The first is in the area of retinal amyloid imaging, which many of you know about. It's really the tip of the spear of what we've been doing the last 10 years. It's directly imaging amyloid in the retina, pursuant to work that was done by Cedars-Sinai Medical Center of this initial discovery and correlation in human cadaver tissue of retina and brain and amyloid. We have 24 devices in studies around the world. We have 2 of our 3 510(k) clearances, one for the device itself, one for an automated software as a medical device tool chain that we call Athena, a fully automated way to analyze these images that is now 510(k) cleared by FDA. And then we have a third where we're doing validation studies in 8 sites, 4 in the US and 4 in the UK. We're getting very promising results in our internal validation studies. And of course, we've had some delays in collecting data due to COVID, but these sites are now starting up. What people are not really so aware of is the work that we're doing in blood biomarkers. This has been underway for several years now, and we've assembled really a very, very seasoned team that goes way back in blood biomarkers with respect to early days work. But this is all pursuant to an agreement that we have with a company in Silicon Valley that has a very high sensitivity platform, and it can multiplex multiple biomarkers. It's very much like a classical ELISA, but it's based on magnetic nanoparticles that generate a magnetic field that induces an electric current so that you can basically, with sensors that can be used. So I'll hold one up to the camera here. This is what a typical PCB board looks like, with each of these fingers on the end that contains 80 sensors that are each individually pre-functionalized with various targets. So this gives us incredible multiplexing capability, ultra high sensitivity from attomolar, to sub-picogram per ml sensitivity. And we feel that it's really a combination biomarker approach that's going to help us solve this ultimately long-term, not just with respect to detection of amyloid positivity, but longer with respect to looking at disease progression and response to therapy. And so those are the main aspects. We get asked a lot, do you have to put retina and blood together? No, we don't. We have done some preliminary work, and we do improve the area under the curve. And it becomes very interesting when you think about the FDA label and ultimately long-term having a test with very high negative and positive predictive value. But these things will be independent. We'll let the data decide, but we've made very good progress in our blood biomarker program with a proprietary amyloid test that's showing very promising results for prediction of amyloid positivity, and in our ultra multiplex, where we're putting together in a single assay amyloid beta 4240, 3 flavors of p-tau, 181, 231, 217, a few flavors of total tau, GFAP, and neurofilament light. Best way to think of it is almost like an ATN chip that can ultimately be used not just for cross-sectionally, but looking at patients longitudinally. So we're hopeful to be bringing forth a laboratory-developed test to market later this year, and ultimately we have a multinational collaborator to take this all the way through IVD, and I know you'll be talking about scale later. But in a nutshell, that's what Neurovision is, and very excited to be participating in this roundtable.
Thank you, Steve, and very interesting. And now we have Michael with us. So Michael, can I ask you to introduce yourself and the technology you represent?
Sure, thanks, Steve. Thanks, Jeff. So I'm a professor at the University of California, San Francisco, and I'm principal investigator of the Alzheimer's Disease Neuroimaging Initiative, which is a large project involving over 2,000 participants, which has been longitudinally following cognitively normal, mild cognitive impairment, and people with dementia for 15 years. And the overall goal of this project, called ADNI, is to validate biomarkers for Alzheimer's disease clinical trials. We tend to think of two continuums, which are both going on in parallel. There is the biological continuum, where we conceptualize Alzheimer's disease as amyloid plaques, tau tangles, leading to neurodegeneration, and accompanied frequently by co-pathologies such as cerebrovascular disease, alpha-synuclein, and TDP-43 accumulation. At the same time, there's a clinical continuum of people who are cognitively normal, people who have mild impairments, and people who have dementia. And these two processes go along in parallel, but they're not always completely aligned. So completely normal people can have quite advanced pathology, for example, and there are people with a lot of dementia who don't have Alzheimer's disease pathology. The big question going forward is, what is the best biomarkers that we can use to identify the pathology? Up until now, we're using primarily amyloid PET scans and tau PET scans to measure amyloid and tau, and MRI to measure neurodegeneration. And cerebrospinal fluid measurements from lumbar puncture are also useful to identify people with amyloid in the brain. But only amyloid PET can really demonstrate that plaques are removed by treatments, and that's very important, as we've seen from the recent Biogen approval. The problem is these PET scans and MRIs are quite expensive, and at least in the United States, they are not reimbursed by insurance. The excitement is the development of blood tests, also for amyloid, tau, and for neurodegeneration. And what we're all trying to figure out now is, how are these blood tests going to be used in clinical trials? And ultimately, how are the blood tests going to be used in clinical practice?
Thank you, Mike. I look forward to more in the dialogue. Michael?
Thank you, Jeff. It's a pleasure to be here among this esteemed panel. My name is Mike Larazari. I'm Vice President of Clinical Research for the Neurology Business Group at Eisai, and I'm responsible for the clinical development and overall strategy of Eisai's neuroscience portfolio, which includes neurodegeneration, epilepsy, and sleep. By way of background, I'm a neurologist and I've worked in Alzheimer's disease for over 25 years, including basic research on animal models, biomarkers, and clinical pathological correlations, and therapeutic drug development across all stages of drug development. As you've been hearing, and as you will hear, we are really entering a new era of Alzheimer's disease with rapid advances in the understanding of the pathophysiology of Alzheimer's disease and the development of biomarkers that can detect the changes of Alzheimer's disease in the brain, often years before the onset of symptoms. And these advances are incorporated into Eisai's portfolio strategy. Eisai has a deep portfolio of molecules in development that target amyloid with lecanemab, tau with an microtubule binding domain region anti-tau antibody that's preparing to enter phase 2, and neurodegeneration, so a phase 1 molecule that targets neuroregeneration and synaptic regeneration of the cholinergic system. Eisai is also collaborating with diagnostic companies such as Cogstate and Sysmex to help advance diagnostics to detect clinical cognitive impairment, biomarkers of disease, and also facilitate diagnosis and care of individuals with Alzheimer's disease. Thank you.
Thanks, Mike. Bruce?
Yes, good morning, good afternoon, and again from my side, thank you very much for inviting me to this panel, also with all these esteemed colleagues. So my name is Bruce Jordan. I work at Roche, and I mean, Roche clearly shares the vision that this community has to really detect Alzheimer early before irreversible damage is caused, and then be able to treat that disease in order to make sure that you don't lose the person who you are. And I'm from the diagnostic side, so I'm the international business leader for personalized healthcare solutions in Roche Diagnostics in Rotkreuz, Switzerland. And my area of focus is really on the fluid biomarkers. So we have various different technologies within Roche Diagnostics. And I would certainly like to acknowledge that whilst we work on fluid biomarkers directly, imaging, digital tools, clinical assessments, etc., have a huge role to play together with the biomarkers, the fluid biomarkers, in order to improve the patient journey. So specifically in Alzheimer, we have, I would say, 3 areas of focus. The first is on the spinal fluid assays that are available today in the countries that recognize the CE mark, so amyloid beta 42, phospho-tau, total tau. And we also received breakthrough device designation from the FDA for those assays and hope to have them available there soon. So that's one area of focus. The other area, which has been mentioned many times, and I think the science here has been fascinating to see this evolve over the last few years, is in the blood-based biomarker space. So this is something we have a very keen interest in the research and development field, in particular with our academic collaborators. So we work with the leading academics in this space to really keep us at the cutting edge of where that science is going. And I think the third thing that I just wanted to bring to the table is a really nice concept or collaboration model that we call the Neuro Toolkit, which is also together with many academic leaders, clinicians, as well as industry, philanthropy. So we work across a broad number of different stakeholders to really try to understand where is the diagnostics development going next. So how can our tools help understand the biomarker science, but in particular be able to translate this into routine clinical practice, both with the CSF assays that we currently have available today, as well as what we bring through our pipeline has access to a huge installed base of active analyzers that are already in situ in laboratories throughout the world. So for us, it's really that scalability, I know, is on the next part, but those 85,000 analyzers generate greater than 15 billion test results per year. And we use the same analyzers for all our assays. So we have real scalability there in terms of being able to deploy these new innovations into clinical practice. I think also from having both the diagnostics and the pharmaceuticals units under one roof, we also look maybe a little bit more holistically at the patient journey and how can we improve, how can we unlock roadblocks, certainly in the diagnostic journey, and if necessary, combine with imaging technologies or with digital technologies or PCR technologies, whatever works best, because I think we're all faced with the huge challenges of biomarkers in blood, with analytical sensitivity, with the ability to detect these low abundance proteins and molecules in blood. And I think we've got a really exciting time to look forward to in the coming years. And we really look forward to collaborating further with the community, because this is a community of work that I think really brings the field forward. So with that, I would again say thank you very much. And yeah, I look forward to the lively discussion later.
Thank you, Bruce. Gert?
Hello, everybody. I'm sorry, did I unmute myself? Yeah. Am I still muted?
I can hear you.
Okay, sorry. So thanks for the invitation to participate on this panel discussion. So I'm Gert Jannes. I'm responsible for the business development and OEM partner management at Fujirebio Europe. We're actually a member of the Fujirebio group. Fujirebio is active in the development of in vitro diagnostics, and we develop assays for routine as well as novel biomarkers in different disease fields. But with the acquisition of Genetics in 2010, actually a real pioneer in Alzheimer's diagnostics became part of our Fujirebio group. We actually introduced CSF biomarker assays already more than 20 years ago with our manual InnoTest assays for the core biomarkers in Alzheimer's, being amyloid and tau. And these assays have been used and are still used today in a lot of research studies and clinical trials, and also in vitro diagnostics. However, recently we have actually developed new assays on our fully automated immunoassay platform, which is called LumiPulse. And we did this because we believe that access to reliable and automated biomarker assays is really what we need if we want to scale up biomarker testing into routine clinical care for Alzheimer's disease. So today, these assays are already CE IVD marked, and they are being used in Europe for diagnostics and even in several other countries outside of Europe. We made them also available in US in CRO labs. They have been completely validated for use in clinical trials. But also in November last year, we actually submitted our data for the CSF amyloid ratio to the FDA. And the amyloid ratio has consistently showed actually high concordance with amyloid PET, and therefore our intent to the FDA is to propose this amyloid ratio test as an alternative for PET scans. Next to that, of course, looking at the future, with the progress being made on blood biomarkers, we are also developing our own pipeline of plasma-based markers on our current platform. And our first priorities are actually focused around beta 181 and neurofilament light. Next to that, like we did in the past, the development of new biomarkers is of, I would say, of utmost interest of us. And we have several ongoing programs in-house to look at novel biomarkers. And we do this either by our own programs or by collaborating with external parties. So it's clear that for Fujirebio, Alzheimer's and neurodegenerative diseases is really a key area for our company. And I look forward to have a good discussion today.
Thank you, Gert. Joel?
Jeff, thanks so much. I appreciate the introduction, and certainly on behalf of C2N, we're delighted to be here today to be able to talk a little bit about our company and participate in this discussion that's more important than ever. So C2N Diagnostics is a company that I co-founded with a couple of colleagues, including Dave Holtzman and Randy Bateman, during 2007. So you could ask the question, what have we been doing for the last 14 years? It's been a lot of knowledge accumulation and understanding what we think will ultimately prove to be the most robust methodologies to be able to track disease pathology. And so our platform is based off of mass spectrometry, and we can use the mass spectrometer in a number of creative ways. One, they're obviously highly precise instruments, which certainly helps in the case of analytes that have narrow dynamic range, which in the case of amyloid can be an issue, particularly when you're sampling blood. And then in addition, the ability to multiplex. Obviously, that's been an important topic of discussion because, you know, we, like others in the field, recognize that there likely isn't a single biomarker that's going to tell us everything that we need to know about Alzheimer's disease and certainly other forms of neurodegeneration. So we take this platform, mass spectrometry, and we've done extensive automation and validation of that technique platform over the years. And in 2016, one of our academic collaborators, Randy Bateman, who's well known to many people in the field, was able to demonstrate that you could use high resolution mass spectrometry to identify significant differences in the way individuals who have amyloid pathology versus those who do not. And we took that test and we kind of automated it, scaled it up, industrialized it. And I'm pleased to say that in 2020, late 2020, we introduced the test Precivity AD, which is a blood test that leverages several analyte measurements, amyloid beta 42 and amyloid beta 40, and presents it as a ratio, combined with apolipoprotein E proteotyping, effectively the equivalent of genotyping. And we can provide what we call an amyloid probability score, which would be indicative of the probability that an individual has amyloid in the brain. And our correlation studies have been extensive, both with cerebrospinal fluid as well as with amyloid PET. We do have a breakthrough device designation with the FDA for developing this blood test, you know, as a in vitro diagnostic that is recognized by the FDA. We also have a CE mark, and so our current activities are focused on, as we're getting vitally important information from the clinical community, because this test is now being used in the clinic for clinical decision making to help physicians better diagnose Alzheimer's or rule it out. We're doing extensive academic collaborative work to understand how well this marker and additional markers that we're working on can identify disease at the earliest stages, differentially diagnose from other conditions that can masquerade as Alzheimer's, and then track the disease and ultimately be able to also measure responses to therapy. So all those activities keep us busy, and I'll be delighted to continue the discussion as this conversation continues.
Thank you, Joel. Thank you to all the panel members. If I could have everybody have their camera on now so we can have a discussion. I am going to direct some of these questions to individuals, but I would be delighted to have an interchange here and have anyone comment on the questions that I'm raising. Mike, I'm going to start with you. The approval of aducanumab was based on a biomarker reasonably likely to predict a clinical benefit. Are we in a position to have other biomarkers that have reached that state?
I think at this moment, the answer is no. I think the aducanumab approval was based on the fact that the treatment removed amyloid plaques. There are a number of diagnostics that can detect amyloid plaques in the brain, which would include obviously amyloid PET, as you can see the plaques, cerebrospinal fluid of amyloid because the A beta 42/40 ratio goes down in the cerebrospinal fluid. And that's been shown in our study and others to be a very valid predictor of the amyloid PET and pathology. And then there's blood that we've all been talking about, plasma amyloid and species. But when you're talking about clearing of plaques, to my knowledge, the only modality which can convincingly demonstrate plaque clearing is amyloid PET, or unfortunately autopsy. The cerebrospinal fluid measures and the plasma measures at this time have not been demonstrated to demonstrate clearing of amyloid.
Thank you. Thank you. Any other comments? Joel, I was wondering whether you think that the serum or plasma amyloid measures will reflect the decrease in amyloid in the brain. I don't think we have data on that yet, but I wonder what your thinking is about it.
Yeah, so we do expect data like that to emerge. How robust that data is and exactly how predictive will obviously depend on the data. But we do believe that with an adequately precise tool, and this is again one of the virtues of mass spectrometry, you can pick up very subtle distinctions in what's happening to the biology of the analytes, in this case, amyloid beta. So in our assays, we've been able to, we actually report out a quantitative measurement, and that quantitative measurement is actually, it's intentional because while there's certainly value in having a report, for instance, in the case of amyloid PET scans in the clinic, oftentimes physicians receive a report from the radiologist who says positive amyloid, negative amyloid. But we know that patients ask the question after that, well, how much amyloid? And so having quantitative measurements, which has been proven useful in LDL cholesterol lowering, and hopefully we're embarking in a new era of disease modifying therapy where it will be important to track the analytes that are being implicated or intervened upon by the therapies. And so when we develop our quantitative measurements, we are innovating with that concept in mind. This is an area obviously of intense focus. We have multiple large investigations ongoing that are testing multiple novel interventions and what happens to our blood-based biomarkers. But we do think the quantitative elements of what we report will be useful, and ultimately, we're confident that there'll be promising data to show that one might be able to use our marker as a disease-modifying indicator.
Great,
Jeff,
thank you. I can't wait to see those data. Yes, Steve.
Yeah, I think to amplify a little bit of what Joel said and also what Mike said, yeah, where we are today, we don't necessarily have a marker that looks at amyloid reduction per se, but I think we all step back and look and realize we live in an age of data science where we're going to have multiple markers that might paint a picture of what progression might look like that correlates very well with that. And so embracing this approach that Joel has just mentioned with respect to looking at multiple markers, creating a biosignature of what someone who is progressing or responding to a treatment looks like. Is their p-tau going up? Is their neurofilament light continuing to go up? Are we bending the curve? Getting that longitudinal data is going to be very important. At the end of the day, though, it's clinical decision-making. I mean, I see our role and others' role is augmenting clinical decision-making. There's going to be multiple factors that clinicians are going to have to look at to try to make a determination due to the multifactorial nature of this disease and where it's born from. So I think that a biosignature approach with multiple biomarkers really will, maybe some will paint a picture that really strongly correlates with what a PET scan can show, but perhaps multiple biomarkers will inform clinicians on good clinical decision-making on treatments.
Great, thank you. Gert, I'd like to hear about CSF and the role CSF would play in this dialogue, both diagnostically and therapeutically.
Yeah, I think, of course, if you look to, I would say, the role of CSF today, and then we look to fluid biomarkers where we are, then I think we have to say that at least CSF biomarker testing has proven some validity and has really reached what I would call a validated status. It's really highly concordant with amyloid PET, and I think it's a perfect alternative, like Mike already mentioned in the beginning. And for me, there are three things that actually are really been achieved for the CSF biomarker testing that's very important. And that's on the one hand, I think the access to fully automated platforms. But secondly, also the fact that we have established some certified reference materials, which really would allow to get really universal cutoffs, which can be used all over the globe, and people will start testing on a larger scale. And thirdly, I think what is also very important is the fact that also for collecting and handling CSF, there have been established some international guidelines. And I think these three things will really allow to look at CSF biomarker testing as a real, I would say, good alternative for this amyloid PET today. And I can speak from our experience in Europe. In Europe, actually, CSF collection is really widely spread and very well accepted by patients and healthcare providers. And it's also, I would say, picking up very well in the US today. And patients should not be scared of taking a lumbar puncture. And this is really a safe and a well-established procedure. So I think today, CSF is surely there to stay, I think, as an alternative for amyloid. And to my opinion, it's also there to stay when blood biomarkers will come into play, because today we're still not sure about, I would say, what will really be the prediction of this blood biomarker. But of course, if you want to move into the general population and think about screening larger groups of patients, then we need to move to blood biomarkers. But this can maybe be seen like a kind of triage system or a pre-screening system where we can identify those patients in which we say, okay, we will bring these patients to another kind of level of diagnosis where we have amyloid PET into the play or CSF biomarker. So I think we really have to look at some kind of complementarity to all these diagnostic tools that we are developing and other biomarkers like Stephen already mentioned, to really get a nice picture on what's happening with the patient when you look at him.
Jeff,
Yes,
can I just,
please, Mike.
I just briefly interject? I just want to follow up on what Gert was saying. I think in the United States, or at least in North America, there's an opportunity to amyloid phenotype more people by using lumbar puncture and cerebrospinal fluid measurements. We only have so many PET scanners, and the demand on these PET scanners is just going to go up a lot. And I foresee the establishment of clinics where lumbar punctures can be done on scale, perhaps by nurse practitioners. It's a very reliable way to identify people with amyloid pathology who could have treatment and who can enter clinical trials.
Great, good point. Bruce, I wonder if you would give us your views on CSF in a world where blood and imaging are also playing important roles diagnostically and therapeutically.
Yeah, sure, Jeff. I mean, a lot of these technologies and tests have a role to play, I think, in multiple parts of the patient journey. And I think it's been mentioned before, the interplay between these different technologies and how they enable clinicians to really effectively manage those patients. I think we take a step back and we look at it starts with a healthy person that, you know, wants to know maybe what their risk is of developing certain conditions. You have people who may well be having the biochemical signs of disease, but it hasn't yet come out into clinical manifestations, all the way through to people on treatment who may want to see how their amyloid is being affected in the brain. So I think there's really an enormous amount of opportunity for amyloid treatment to be even better influenced by diagnostic tools, be they in vitro or in vivo imaging. And I also think this depends really on where you are in the world. So yes, in Europe, for example, spinal fluid testing is routine. Imaging is also widely available. In the US. Maybe there's, to your point, Mike, there's something we can do to help really scale things up. And then if you go to the other extreme in China, there is neither imaging nor, at least the PET imaging, nor the spinal fluid testing really available there. So that certainly may be a place where we might have to think very differently about what is needed in China in order to be able to help people move through their journey with dementia. So that's, I think, we do know, and even within different countries within the United States, I imagine there's a huge difference regionally in terms of how, how each state manages those patient journeys. So I think all these technologies have a role to play. They likely have a role at multiple stages in that patient's journey. And I think with the blood test coming through, to be honest, a few years ago, I didn't even think we would seriously be thinking about this with the the limitations of technologies from years ago, and the fact that we're actively discussing this as a reality that thanks to C2N is already happening in the US, and all the great publications that have been presented over the last 2 to 3 years that have shown that blood biomarkers have a huge potential to play, and not only in one part of the journey, but maybe there's multiple different places where people can improve their diagnostic journey, and then their hopefully their improvement based on treatments. So I think it's really exciting, and I think we've only just seen the start of where this is all going to land.
Thank you. And that global perspective, I think, is so important. Mike Larazari, I want to turn to you. I know ASI has been in the forefront of digital biomarkers, and I think it's important to have that as part of the dialogue as well. Could you tell us a little bit about what you're doing there?
Sure. At ASI, we think that digital tools, especially digital tools to assess cognition and function, provide the opportunity to support brain health, as well as early screening and detection of cognitive changes. An example that I'll give is our collaboration with Cogstate in Japan, a platform called NoKnow. So in Japanese, know means brain, and then in English, know means to know your brain. And NoKnow takes an established computer and tablet-based cognitive test from Cogstate, which is used for clinical trial screening, and which is also used in clinical practice for screening, the Cogstate Brief Battery and the Cognogram, and offers it to individuals by website with a focus on brain health. Individuals have the opportunity to take this cognitive test, and the results are provided back, as well as information regarding improving their cognitive health, and also providing an opportunity for individuals to discuss the results with their physicians. So ASI has been collaborating with municipalities and corporations to include brain health, community-based screening, and annual health checks to facilitate people taking charge of their cognitive health as well as their general health. And these, more globally, ASI is working with Cogstate in terms of implementing the cognogram to facilitate earlier diagnosis and earlier screening in clinical practice as well.
Great, thanks. Stephen, you were talking about the interaction of the needing to make the clinical diagnosis, that's still a clinical event with a synthesis of different types of information. How do you see this dialogue between the cognitive testing that we've been hearing about from Mike and the evolving biomarkers? How do you see one complementing the other?
At the end of the day, actually, I think that the FDA will be the ones that mandate this, because it's what I've learned, one of the things I've learned in ophthalmology, it's very difficult to have a biomarker, a measurement, a tool, and directly link it to a diagnosis of a disease. And it always comes down to supplying a clinician with valuable information that they synthesize together and put that picture together. So in ophthalmology, it's a visual field, it's an OCT. In our domain, it's cognition with blood biomarkers with MRI. I think that as time evolves, some of these tools will give better predictors by combining them together to inform clinicians on how to make better decisions. I don't see any time in the near future where we have AI that is making decisions for clinicians, to be very clear. But I think that the tools will evolve so that you can take data sets that are orthogonal to each other. So for example, we've taken blood and retina and put them together, and we get a 5 to 10 point jump in area under the curve. I think Oscar Hansen in in Lund did similar things with p-tau and cognition to come up with different predictors, not for amyloid positivity, but towards the onset of Alzheimer's in 4 years. I think that these tools will evolve. I think that they won't make their way into the clinic in terms of actual, decision-making, but it'll augment decision-making, allow clinicians to integrate that information. And the easier we make it as companies to integrate that together so that they can have those clinical dashboards to make those decisions, then the more informed they'll be in differential diagnosis of really understanding what's the best course of treatment for a patient or a change in course of treatment for a patient.
Thank you. Joel, I wanted to turn to you about globalization. I think there is a concern about equity, both in terms of diagnosis and therapy. So you're pretty far down the line here with your development. So how do you see globalizing the Procivity test?
Yep, great question. Thanks, Jeff. So I think there's, this feeds upon some of the other comments that have already been said. So, when we look at the precision, power, resolution capabilities of mass spectrometry, and we say, great, that's a terrific platform. But to actually have a global impact, you have to be able to take that process and be able to automate it as much as possible, standardize it, and then ultimately have collaborators who you can rely upon and trust, who will be part of this effort of globalization. So when we look at like what we're doing at C2N, we've already done a great deal of kind of optimization of the assay, where we can now, a single instrument to be able to run these analyte measurements will put out 30 to 40,000 tests a year. So it's not a question of the feasibility of reproducing, a mass spec to be able to run through high volumes, but it's thinking about the scalability. So one of the things that we're doing is we're actually we have a big project underway at C2N where we are looking to ultimately qualify other exceptional reference labs throughout the world who will effectively be partners with us, and we will have a proficiency service element, a collaborative element whereby we're qualifying these labs to be able to perform the Procivity AD test and other tests that will be added to the panel in the near future. And that's an important step towards globalization because, we realize as a specialty lab in St. Louis, it's hard to accept samples from, Japan or South America without certain logistical complexities, notwithstanding even the costs. And so we expect to have a really innovative program with other reference labs where we will be qualifying and we will be providing qualified reagents and components to standardize. I think Bruce did a nice job talking about the idea of reference standardization and certified reference material. We see mass spectrometry as being able to play an enormous role in that. And so those are some of our efforts. I think the issue of the accessibility and minority underserved populations is a huge issue. And to be honest, the existing tools with PET scans and CSF, given the specialty requirements, there hasn't been enough effort to make sure that this is accessible to people who live 100 miles from the nearest tertiary care center. So the ability to put this in the hands of primary care physicians, and that's ultimately where this field is headed, because if we really want to impact the disease, we are very strong believers in this concept of prevention, which means you have to intervene before symptoms are onset. And this brings up the critical necessity of having dynamic, resolvable biomarkers that can pick up disease 10, 15 years before onset, which will then put in place models throughout the world so governments can start focusing on individuals while they're still healthy to be able to maximize their independence and functionality. This is a huge effort underway and a huge priority for the company to understand that better and to be able to provide part of the solution.
Thank you. Stephen, did I see your hand go up? No, maybe not. Okay. Gert, I'm going to give you the last question. The time has gone so quickly. What about new biomarkers? There are so many biological processes that we're not measuring. I'm frustrated by not having a good inflammatory biomarker. So how can we stimulate new biomarkers to measure more biological processes?
Yeah, I think what is important if you look at new biomarkers is really I would say the level of collaborations that are ongoing. I think there are two key players, I think, also in the field. That on the one hand, you have the academics and the pharmaceutical industry who can learn us a lot, I think, about the targets, the biological targets, the pathways they're looking at. When looking at your recent publication, Jeff, about the new drug development pipeline, I remember two things. There was a conclusion that said, okay, the targets are becoming more diversified, which is clear, showing the need for more biomarkers. And the second conclusion was biomarkers are being more regularly used. So I think there is really this need for novel biomarkers to be developed. But of course, you can develop an assay in a research format, but then you still need to prove the clinical value. And for this, I think the second collaboration is very important. That is access to samples, to really well-characterized samples, which are coming from a longitudinal follow-up studies, and then organizations like APNI and organizations that really are able to share well-characterized samples, which will allow us really to test these novel biomarkers. I think these two things are, in my view, key, I would say, and they're all bundled, I would say, in one word, and that I think there should be a good collaboration between diagnostic companies and pharma industry, academics, and people that are doing large-scale studies.
Thank you. Terrific point. Well, I'm going to thank the panel members for all of your contributions. I wish we had more time. There are so many interesting questions I would love to ask you, but we have to go on to the second panel. So I'll ask you to turn your cameras off, and I will ask panel 2 to turn their cameras on.
Your phone's off.
Yes.
Great. And in this panel, we're going to be discussing opportunities and barriers to Alzheimer's disease diagnostic adoption and scale. And again, this is a realm of innovation, which is so important. So we're going to start with a round of introductions and discussions of the technologies of each of the person, or I should say maybe the realm of activity of each of the people. And John Dwyer, could we start with you?
Sure. Thank you very much, Jeff, for inviting me, and to High Lantern and CEOI. I'm John Dwyer, the president of the Global Alzheimer's Platform. We are actually a technology agnostic enterprise trying to deploy technologies for reducing the time, improving the quality, and reducing the cost of Alzheimer's clinical trials. Thank you.
Steve, let's go ahead with you.
Well, hello, everybody. My name is Stephen Smith. I serve as Senior Vice President and Chief Scientific Officer at Advent Health. We're a very large healthcare provider with over 50 hospitals in the United States. And as a faith-based healthcare system, we're committed to whole person compassionate care and focused on using the best science to guide our clinical practice. We'll talk a little bit more about that in a few minutes. We feel like we have a moral and mission obligation to learn from our patients and consumers so that we can provide the best care. And clearly here in Florida and the US, Alzheimer's and brain health are top of mind for so many of our consumers and communities.
Thank you. Tony?
Sure. Yeah, thank you for inviting me to this esteemed panel. So I'm Tony Fair. I'm the Chief Medical and Clinical Transformation Officer at Highmark Health. So our organization is an integrated payer provider system that's based in Pennsylvania with 4-state regional reach, but also national reach across all 50 states. And our strategy, which we could talk about some more, is focused on improving health outcomes at scale. There's a lot to talk about that, but the basic principle is that 30% of the healthcare dollar is considered to be waste in the system for a multitude of reasons. And we've proven over the last few years that not if, but when you improve people's health, the total cost of care comes down. Emphasize the word total. And that can be reinvested in in bending the cost curve, but also in reinvesting in the capabilities that allow us to do the sorts of innovations that we're talking about in this dialogue.
Thank you, Tony. Beth?
Good morning, and thanks so much for having me here today. My name is Beth Roberts, and I'm a partner in the healthcare group at Hogan Lovells, which is a global law firm with over 500 of us focused just in the life science area. My practice is really helping both diagnostics as well as therapeutics and imaging agents get Medicare coverage and reimbursement. I work really closely with trade associations and patient groups as well as manufacturers of the various diagnostics and devices. And I also have a really strong personal passion in this area, as I had a mother who was diagnosed with Alzheimer's disease and unfortunately passed away from the disease in 2017. But this is very much a personal journey that I've been on and know how important it is to get a diagnosis and ideally treatments here in the future.
Thank you. Jason, could we hear from you?
Sure. Greetings. Hi, Jason. Jason Carlos, joining you from Philadelphia. I'm a professor at the University of Pennsylvania. So the perspective I'll offer on opportunities and barriers to diagnostic adoption and scale, I'll take from the perspective of the individual, that is to say the person getting the test or choosing whether or not to have the test. The work that we've done, the studies to understand that, show that the decision to get this test, a diagnostic test, is closely linked to the perception that there's some benefit to come from it. No surprise there. What's interesting are what are the benefits that people would like. The studies we've done so far are from persons who have enrolled in clinical trials, so they're getting a drug, albeit not yet proven to have an effect, but certainly the opportunity to get a therapeutic effect from a drug is one reason. But people also talk about other benefits. These include planning for the future. People are quite mindful about how much remaining time they have left of what they will often describe as productive time, however they conceive that. This ties into issues of employment, location of where they live, and their resources, particularly financial resources, in anticipation of potentially a long period of disabling cognitive impairments that will need caregivers, money, and a living location that will work for them. The downsides are notable and largely revolve around matters of stigma. People are quite concerned about stigma.
Thank you. And Jim, could we hear from you?
Thank you, Jeff. I'm Jim Taylor. I'm an FDA-appointed Alzheimer's patient advocate, and this is my wife and daily inspiration, Jerry. We want to applaud the forum for including the voice of the patient, the person living with Alzheimer's, and the care partner. We're often asked our ages. I'm 76, Jerry's 78, but we will not require the rest of the panel to reveal their ages. Jerry was diagnosed with mild cognitive impairment, MCI, in 2012. And since that time, we have spoken to over 10,000 people across the US, in Europe, and now in Japan. We speak to address the stigma of living with dementia, the possibility of living with hope and purpose and joy after diagnosis, and strongly encourage participation in clinical trials. Our current activities is the development of a new pilot program in New York City called Memory Advocate Peers, MAP, which trains former dementia care partners to be advocate peers of individuals who are newly diagnosed with dementia and their care partners. We are partnering with Caring Kind in New York City in this pilot, and the volunteer advocate will bring the newly diagnosed individual and care partner sympathy, emotional support, information about their disease and how to live with purpose, and importantly, personal support in finding a clinical trial. Thank you.
Thank you, Jim. Thank you, Jerry. And thank you both for being here and for your bravery for being with us on the panel today. We really appreciate your courage. If I could have all of the panelists on camera now, great. And again, we're going to do it like we did the last panel, which I'll direct questions to an individual, but I would love to hear from anybody who has a view on that particular question. So feel free to chime in as we're going along. Beth, I'm going to start with you. There are so many questions about reimbursement. And of course, reimbursement is so key to attracting people into the innovation of biomarkers. So could you talk a little bit about how we should be thinking about reimbursing biomarkers, and are there strategies that should be used? Tell us your thoughts about this.
Sure. Well, I will say first and foremost, this has been a really, really challenging area. So the FDA is quite backed up today because of COVID. They really have put all hands on deck, to try to get the COVID tests approved during the public health emergency. The agency now is moving people back to their departments. But unfortunately, that backlog remains. So most of the time when people are going to make a submission to the FDA, they actually go in and meet with a pre-submission meeting. Folks are being told today that really, other than COVID tests, you won't be able to have a pre-submission meeting till the end of the year. And even after that, they have over 5,000 of these approvals pending, if you might imagine, and typically only handle about 2,200 a year. So they are really seriously understaffed for the job at hand here. And clearly that's a concern across the board, not just for Alzheimer's, but as you might imagine, as we heard about all the exciting new innovations in the first panel, to bring those to market, ideally you would be going to FDA and getting approval. There is another mechanism though, and that's the LDT, or the laboratory developed test mechanism, that does allow tests to come to market. But unfortunately, those really are a single lab where the test is performed in a single location. And again, the really exciting innovations that were discussed in the first panel, most of those, I think we contemplate being tackled, across the country, across the globe, and would be things that ideally would go through the FDA for approval. So those are all really the challenges on the FDA side that then create challenges for reimbursement. Because when Medicare and other payers decide whether something is reasonable and necessary, they like to look at that FDA labeling and the FDA assurance and stamp of approval. Unfortunately, ADUHOME was not approved with any companion diagnostic. So, in the actual labeling of ADUHOME, it does not say that there needs to be beta amyloid confirmation. If that were in the label, that would kind of jump it to the top of the line in the FDA's priority list and make it a little easier to argue to payers that these tests should be covered. I do think, though, from a patient perspective, most patients are going to want to know that they have beta amyloid before they, are taking a drug that reduces beta amyloid, right? And I think many physicians will also be looking for that comfort. So the testing is really imperative to identify the right patients here. Even though for the lab developed test, so let's assume this is being performed in a single lab, there are mechanisms to go forward and get coding, coverage, and reimbursement. The American Medical Association is responsible for creating codes, and they have an expedited quarterly process for proprietary laboratory analyses. And so that would mean that if you did have tests and could apply and get a code pretty quickly, And I do think trying to make the case to payers that they should be paying for this testing, perhaps, that really can be made with the argument that this really is to identify the appropriate patients to receive treatments. And, certainly Jason has made a tremendous case that is incredibly important, even if you weren't considering treatment, it's just very important to know. I think for payers, they're going to want to see that more concrete evidence of why they should be paying for this testing.
So that's a great bridge, I think, to Stephen and to Tony, who are both in the position of deciding how to integrate biomarkers into healthcare systems. So Stephen, let's start with you. What's the decision framework for someone in your position to decide on whether or not a biomarker will be used and paid for in the system?
It's interesting. We heard just a moment ago the concept of combining multiple modalities. And we see that as really the future here, the integration and the overall effectiveness of a early screening for diagnosis, and ultimately being able to channel positive patients into the appropriate next diagnostic step. So we're certainly bullish on biomarkers as part of the overall solution, but they're one of a chain of events that needs to happen for a healthcare system to be ready. Historically, the decisions around specific diagnostic tests and biomarkers in other areas has been driven by the individual, if you will, vanguard physician. We certainly appreciate that model and the utility that it's had, but when you're a big healthcare system like us, we need to make these decisions so that they can go to scale very quickly. And so that's why we see digital solutions, not only from a consumer-centric digital solution perspective, but also the overarching system that can stitch all the components together into one seamless whole. So I think it really is going to be, from a healthcare system perspective, decision-making about the entirety of the system. Try, fail, repeat, try, fail, when. And that's really how we're looking at that decision-making. Great question.
Great, thank you. Tony, how about Highmark? How would Highmark make this decision?
Yeah, so thanks for the question. So I would say, first and foremost, since this recent approval, I think it's very incumbent upon the medical societies to kind of fast forward their dialogue around providing at least directional input to physicians at large as to what to do, right? Because otherwise it's going to be a free-for-all, and it's going to be, at least our prediction, quite chaotic in terms of what the patients want versus what doctors need to know to make those informed decisions and so forth. So I agree with Steve. We have both a fairly sizable health system, but also a larger payer that's tied to the Blue Cross Blue Shield Association. So we work very closely with the Blue Cross companies across the US, but we're also very tightly linked with CMS in terms of where the discussions and dialogue is around what in this case will be a very high profile discussion as to where to go, as Beth pointed out earlier. But I would say from our standpoint, we base our decisions, since we're both a payer and provider, first and foremost based on clinical evidence of efficacy. Now, in this case, not to repeat this recent conversation, we're still, I'm going to say, a bit in limbo in terms of, okay, how do you take your next physician who's going to be faced with a long line of patients and family members who are demanding, right, to have certain tests, treatment, whether it's diagnostic tests, treatment, et cetera. And that's why I go back to my first point, which is it's really incumbent upon the medical societies to at least provide preliminary direction as to which way to go. Now, I've been, as a practicing cardiologist, I still see some patients to the extent that time permits. I've been involved in appropriate use criteria in the past, and those are not always used judiciously by some, but I don't know that we're going to have any AUCs on this particular issue today, but I do believe that unless and until the societies come together with some sort of, and I think they will, some sort of opinion, it's going to be very difficult, at least globally, to have a better understanding of what to do. And in the meantime, it's going to be an individual type decision, whether it's at the physician, patient interaction level, or as Steve pointed out, every health system, making their own decisions.
Thank you. Jason, I'd like to turn to you. You've thought a lot about patient interactions with tests like this. What would you see as the necessary safeguards for putting new biomarkers into systems and offering them to patients? What do patients need to know? And Jim, I'm going to come to you in a moment to ask your opinion about this. But Jason, how can we safely and humanely introduce new biomarkers into large healthcare systems?
So if the work from the data, which I think is an important place to start, events of the last few weeks noted, the data are that individuals who learn that they have a marker of a neurodegenerative disease generally understand the information and tolerate it. By tolerate it, I mean that's sort of an odd term. It's one that they integrate into their well-being, meaning they don't suffer catastrophic depression or anxiety. And I realize one's responsible, that's great, let's get these tests rolled out there wide. The reason why we know that is because before they learned the information, they were educated over a period of time about what they were going to learn if they wanted to learn it. And number two, they were assessed for their readiness and willingness to learn the information, their psychological well-being. I can't underestimate or underemphasize the importance of that. And sort of a capricious, so let's get these tests out there, could lead to a lot of harms and waste in the system, both harms to individuals and waste in the system. And without going into the details of what was done, that's what was done. Well, now the problem, of course, is that the scalability of these tests in a healthcare system is limited by the professionals who can do that. The good news is the pandemic, which the only copper lining from it is perhaps we all learned that we can use Zoom competently for its equivalent softwares. And certainly, actually, we've tested with colleagues from the Banner Alzheimer's Institute using telemedicine techniques to disclose ApoE results. ApoE is, of course, not a biomarker, but I think there's a lot of analogies to learning a genetic test for Alzheimer's disease, as well as learning a biomarker of that disease. And we successfully used a telemedicine approach, both video and telephonic, to educate people and disclose the results of a genetic test. And although the data aren't yet published, what we've presented so far in abstract shows people do learn the information, and the outcomes of it are generally acceptable from the perspective of minimizing harm. The larger issue that surrounds that, and I won't sort of play lawyer to Beth Roberts, but is the limitations across states of doing telemedicine approaches, given in the United States, the laws around, who can practice medicine where and for what reason and whatnot. But in summary, this isn't just another blood test. Folks, great line from one of our subjects of why people learn this information was, this isn't just a colonoscopy. A colonoscopy isn't going to change who I am. This is my brain involved. So we have to take these tests seriously from the perspective of the patient. This isn't just a matter of choice, it's about care. And how to deliver good care means education and making sure someone's ready to get it, which means access to a competent healthcare professional, not just a static website.
Thank you, Jason. So let's turn to Jim and Jerry. From a patient's perspective, what do you want to know from a biomarker? How do you think about the biomarkers that are coming out? What's your view of this? And have you gotten information from your peers?
From our peers, no. I think Jerry and I are extremely excited about what we heard in the first panel. It's a remarkable time. We hear very exciting things both in the technology and marker. At the same time, we hear very promising news about more therapeutic breakthroughs. BAN 2401, oligomanate, Lilly has an exciting drug. So we see a great deal of information that's going to be available for people living with disease and care partners to absorb. We believe that information reduces the fear of living with dementia. But obviously there is a spectrum of individuals who don't want to know. And then the person who wants to take advantage of every latest development that they can. I think it's important in trying to pick up on Jason's point of educating, which is right on the mark. It's important to, I think, work before we establish what the education procedures should be, that we work with people in advocacy and who have the disease and care partners to figure out what the most effective approach is to share the knowledge that the researchers have with the general public. And I say general public because not only people with dementia today, but so many of our peers who, because of age-appropriate decline, are afraid they have dementia, are going to want to take advantage of these tests as well. And you think, well, maybe that's not appropriate, but we've had several friends who we have collaborated with who, by applying for a clinical trial and getting an amyloid PET scan and finding out they were negative, removed a huge worry over them because they were biologically, genetically inclined to develop the disease. So we don't underestimate the value of these markers, not just to people who are living with the disease who therefore may qualify for therapy, but for individuals who will have negative results. And they need to be, we all need to be educated too, and carefully and effectively educated.
Thank you, Jim. Very thoughtful. So, John, I want to turn to you as head of GAP and running the BioHermes project in particular, which I'm very excited about. How do you see the evolution of biomarkers in trials, and why are you doing BioHermes? What do you think you're going to do with those biomarkers?
Thanks, Jeff. Well, first, to just frame it, acknowledging that there are many different opinions about it, but from our perspective, the approval of the FDA of Aduhelm is probably the most seminal event in Alzheimer's treatment in easily 40 years. I think and all the momentum that creates can't be ignored. And so we would like to point out clearly that the biomarker tests and the digital cognitive tests that are emerging now will have the opportunity to attract capital and undertake a maturation of their business models that were never available before. And what Beth said about reimbursement is part of the equation, but it's also important that health plans and systems like the Distinguished Highmark Program and Advent recognize that LDTs can be used for a physician to make an informed diagnosis without necessarily presenting that LDT to the patient, especially since it's not FDA approved. And we'd encourage a discussion around that, because I think, and I'll come to the BioHermes in 1 second, Jeff, we estimate we have 14 sites in Florida, Steve. We think you guys are going to see 250,000 to 400,000 visits in your system alone that you didn't see last year, trying to understand the therapeutic opportunity that exists today. And that's going to be more complicated by the fact that your physicians are going to be, a little bit conflicted, maybe even more than conflicted, by not diagnosing for aduhelm, but how do you prescribe a off-label use of Aricept or those kind of products when you have an approved product on the market? So the conflicts within the system are extraordinary, and we have to have a diagnostic and therapeutic process that in an orderly fashion gives you a productive trail of data so you can authorize a PET image, whether they're under 65 or over 65, so you get a dispositive picture. But you don't want to do 400,000 of those in Florida. You'd like to do 40,000 of those in Florida. How do we get to that point? So BioHermes is a comparative large clinical trial, which is incorporated 12 different biomarkers, a number of well-known species that are being provided by different partners, and 14 different digital cognitive tests, all of which will be compared to a centrally read beta amyloid PET image and a full genome sequencing and 2 ApoE4 separate tests, because there's some drift in those tests these days. And finally, about 30 to 40% of the 1,000 participants will also get a tau PET image. So we're trying to provide the industry and the field a comprehensive view of how some of the leading, certainly it's not exhaustive, new technologies show every day of blood plasma based and digital cognitive tests interact with each other and against a gold standard beta amyloid PET image, so that we can start triaging our thought process appropriately for both clinical practice and for increasing the likelihood of reduced screen fails in clinical trials. The other thing about the study that's unusual and relevant to this conversation, as has been mentioned earlier, our track record as a field needs improvement in the enrollment of folks of underrepresented populations that heretofore have been grossly underrepresented in trials. So we have set a 20% participation endpoint. That is to say, our study will not close till we hit 20% of African Americans plus or Latinos combined, which is an unusual study design. Interestingly, we think we've probably shot low, because commentators have said perhaps the right number for a representative study when you include Asian Americans is probably closer to 36% non-white Europeans. So we still have room to improve. But this study will give us, we hope, a reasonable signal on where cutoffs might need to be for people of race. There is evidence that differences in race equate to differences at the level of tau expression or beta amyloid 4240 levels consistent with pathology. But that data is soft in blood plasma as compared to CSF. And so as a consequence, we hope to report this study out as early as February. We're already in the field, and you'll be happy to know, Steve, I think we have like 6 sites in Florida doing the study. We are absolutely committed to making all those folks that participate available to the data available, so they go to their physician with a full package of cognitive tests and a beta amyloid image and come into Advent and say, okay, look at all this stuff I got in this study. Now tell me what I should do. And we are, Jason, giving counseling on the 3rd visit if they're beta amyloid positive, consistent with best practice. But the important thing is, I agree with everyone that says this is early on all these other tests. Absolutely it is. But we can't wait, because patients now have hope, but they need to be well informed and they need to be characterized well in both in clinical practice and in clinical trials. And without bringing these technologies to a new level of trust and reimbursement quickly, I fear that most of the folks that Jerry and Jim and we all know that are patient advocates, those lines are going to be very long, and less good medicine is going to be practiced, because there's 300, 400 sites already advertising that they're in business to administer and deliver Aduhelm. And I don't think those are necessarily the very best settings, and certainly not for a uniform treatment. So that's why we did it, Jeff. We're very optimistic about adding insights to the field, and we will certainly share those. All the data from our study will be available to the general public after a certain safe harbor period, so that health systems like Highmark and Advent and everyone on this call will have access to it.
Great. Thank you, John. John brought up the very important topic of minority representation in all of these systems and experiments and reimbursement discussions. I'd like to ask each person to just briefly comment on what we could do to increase both representation in the things we do and equity in the things that are beginning to be provided. Beth, I'll start with you. What could we do to improve the underrepresentation of minorities in both healthcare and in science?
Well, certainly this is a key goal of the Centers for Medicare and Medicaid Services, and yet we all realize what a challenge it is to accomplish. I think it is critical, though, to get more diversity in the clinical trials themselves. And it's really only by doing that that I think then we can also provide that equity when it comes to actually delivering the diagnoses and treatments to patients.
Great. Jason, how would you view this issue of trying to get better representation?
I think we need to ask ourselves equity with respect to what. Right now in my country, America, it's still very difficult to even get a diagnosis of whether you have cognitive impairment and then access to long-term care services and supports. This is only amplified by individuals who have social and economic challenges. I'm befuddled how access to biomarkers will make that more possible, et cetera. It'll just only amplify the inequities, I think. So this problem has existed for a number of years, and I'm not confident that a blood test for a biomarker and whatnot is going to solve it. I think it will only amplify it.
And Tony, how would Highmark approach the question of better representation, better equity?
Yeah, and much like you just heard on this dialogue, this is not just relevant to this topic here. It's a much broader conversation. And so we view ourselves as a convener, because if we just keep doing the same thing over and over again, the equity situation is not going to improve. So we see ourselves as a convener, because until you include proactively community-based organizations and other organizations that people who are in those situations trust and really relate to, it's going to be very hard to achieve any incremental improvement in that space. So we've been proactively outreaching into those communities that we serve, because if we just serve those who have the means, we're just not going to make any impact. So community-based organizations is one. Two, I know we talked a little bit about digital. We've recently partnered with Google and Verily. Verily is a sister company to Google, on taking the initiatives that we've proven have worked, both more globally as well as in those in those underserved areas, and to try to figure out how to reach a much broader population, including the underserved population, and more efficiently and more effectively. There's a lot more we can talk about that topic. It's early in our collaboration with Google, but we see that as an important and complementary step in reaching those populations.
Thank you. Stephen, what's your view, and from the Advent Health perspective, on minority representation?
Well, I think one has to walk into the conversation with humility and with a really good ear. These are challenging times and challenging conversations, and I think we need to listen very carefully as we connect with our communities more deeply. That is at a very high level. At a very practical level, I would agree with Tony. We need data in our hands to be able to understand where the greatest gaps are in this space, and whether they are social, economic, race, whatever those gaps are, we need data to be able to target and to find ways to focus our limited resources on this. And if we don't have data, we're going to be lost. We can listen all day, but if we don't actually know, it's going to be really, really challenging.
Thank you. Jim, I'd like to turn to you. You're right in the battleground here. So how do you think about minority representation and getting more patients into research and into therapy?
Jeff, the program I mentioned earlier, MAP, has a specific focus on serving underserved communities. We will recruit our advocates and our clients using hospitals, Montefiore and Bellevue, who have large minority populations. And we want to match advocates and patients, newly diagnosed people with the disease by ethnicity, if that is possible, if they're positive on that, to develop the trust that Steve and Tony mentioned earlier, because we believe that establishing that trust in more familial, underserved communities is very important. We'll also try to do that through community advisory boards, and our partner, Caring Kind, has a long established and trusted presence in a number of these communities. We may not reach 36%, as John mentioned, but we have a high goal in that regard. And then we would like to get as many as 40% of our clients to a lab to at least consider participation in a trial. So I hope to work personally with all our clients to explain. We believe a reason a lot of people don't enjoy participating in clinical trials is there's so much misinformation. People don't know they're free, they're afraid of the cost, they don't have transportation. So we're trying to address as many of the inhibitors to participating in a trial, both for people who look like us and for underserved communities as possible. And to see and provide, we've got a research component of our study. So we hope that we can provide some of the information that Steve was asking for on what works and what works by different communities.
Thank you. That's great. We're running out of time, but Beth, I'm going to ask you one last question, and the answer will have to be a little bit brief. But one of the disappointments in the field is the failure to get PET reimbursed, amyloid PET reimbursed, and I think that has had an inhibiting influence on the field. Are there specific lessons from amyloid PET that we should be thinking about in terms of other biomarkers or in terms of continuing to work on amyloid PET for reimbursement?
Well, Jeff, I couldn't agree more. And actually, I think it's non-coverage, as well as any potential non-coverage of other diagnostic tests, really exacerbates the disparities that exist today too. Because if only people with money can afford to pay out of pocket for these tests, then they're going to also be the only folks who have access to the treatments themselves. But I do want to point out that CMS has been asked to reconsider the PET national coverage determination. Your voices are really important. And so really one of the lessons learned is that this is imperative to have coverage for the PET scans. And CMS should take this up and should reopen, not just for bridging studies that continue to cover one per patient if patients are in clinical trials, but also more broadly. I think the evidence is there that these tests are reasonable and necessary, and it's time to revisit that. And I do really encourage all of you to become involved as advocates to send that message to CMS.
Great, thank you. Well, I want to thank the whole panel. What a great panel. And the time went so fast. I wish we had more time to ask more questions. But I want to thank you, and we are running out of time. I want to make just a few closing points, things that I've learned as I've listened. One is necessity for harmonization and understanding similar things about all of our biomarkers. So looking forward to creating an infrastructure that would be able to do that. I think the challenges in coverage and reimbursement are so important. They're what motivate the development of new biomarkers, but they also provide the framework for better care and more precision medicine, which of course is our goal. We need to keep patients at the center of our efforts. And Jim and Jerry, I am so grateful for you being on here and taking the patient's point of view and bringing the patient's point of view into this discussion. It's really important. You're what it's all about. So we really want to make sure that we're listening, as Stephen said. And then, of course, we need a support for a global mechanism, and Davos has been very eager to take on the harmonization, the collaboration, the uniting of humanity to try to advance therapeutics and diagnostics. And we've seen a great step forward in the recent past, blood test, imaging, new CSF tests, new digital biomarkers. They're all coming towards us. I'm so excited about the future and integrating them into the care and getting better and better care. So we thank you all from both panels. We thank Davos, and we'll close the panels now. Thank you very much for attending today.